So if we're starting from our first order neuron, the same thing is true. We would have that neuron somewhere in the skin, probably in the dermis of the skin on one of those tactile corpuscles, and that'll have dendrites. The axon will follow through a nerve, depending on where that nerve is. You might have it go through a brachial plexus, the lumbar plexus, depending on where that signal's coming from, or no plexus at all. And then we'd have the unipolar sensory neuron is what we're looking at. So we'd have the cell body in the dorsal root ganglion. And then we would not cross over for this one. We'd go straight up to the medulla, and that entire thing would be our first order neuron. One with a degree after it is just for order, so you could call it first order. You could write that out if you wanted to. Okay, so essentially the first order neuron goes from the dermis all the way to the medulla. It's still going through a tract. It's moving through the white matter of the spinal cord. So then, right at this point, this is our first synapse. So that also means we have the synaptic terminals of the first order, we have the cell body of the second, and we're switching over. There's a neurotransmitter released, all of those things that we learned about synaptic terminals in a synapse to the dendrites of the next neuron. Okay. So then the next neuron will then, in the medulla, cross over. So it crosses over in the medulla and then heads up to the thalamus. So our second order neuron crosses over and heads to the thalamus. So medulla to thalamus. And then our third order neuron goes from the thalamus to the somatosensory cortex. It's okay if you just call it sensory. Sensory cortex of the cerebrum. And like we talked about yesterday, that it'll land on the sensory homunculus. That's a somatosensory cortex of the cerebrum. So what I would encourage you to do when it comes to this spinothalamic versus posterior column tract is, in your studying, compare and contrast. So try and come up with some ways that they are similar, some ways that they are different, right? Both of them are sending sensory information, but the spinothalamic is deep touch, whereas the dorsal column tract is light touch. Both of them are crossing over, but the crossing over occurs here versus occurs there. There are, three neurons that are the same in each of them, right? So think about what's the same, what's different. Make yourself some lists of comparing, contrasting them, because I'm almost certain to ask a question about that in the exam. Okay, so we talked about the homunculus, how we have a homunculus for sensory information. We have a homunculus for motor information. This essentially is full of these little areas that tell us where that signal came from, as well as like the pathway the neuron came on, because it's a specific type of neuron. So then from here, we are going to do a really brief olfactory. So we're just going to go through this pretty quick. Just do the simple pathway. The olfactory bulbs we already saw when we did our practical. They're sort of sitting on the most most inferior part of the brain, really far forward, and they're kind of two individual separate parts, but then they attach through a sinus. So these are the actual bulbs. These are what we saw in our brain. And we're going to see this area of the bone when we start looking at the skull. But this is really what we're looking at, which is inside of the nose. Along the sort of interior of the nose are these areas where we have epithelial cells, and then those epithelial cells secrete mucus. Okay, so that's kind of what we're looking at in this picture. We've got all sorts of stuff that's identified in there. I'm not too worried about the details of that. Instead, what we're going to think about is how we start an action potential. So you smell something, you breathe in, and these little molecules that are odorant molecules move in to the nasal cavity. Move into nasal cavity. And they attach to receptors. And we've got lots and lots of these receptors. Some animals have more. We've lost a lot of ability to attach to those receptors. From attaching to those receptors, they start a G protein signal transduction pathway. So that's that same G protein pathway we've talked about before, where the signal attaches on the outside, moves to the inside. It's activating or making a cyclic AMP. And that's all happening right here. Like, this is where we're still at. It's just at that very beginning. Those are the G protein receptors. Now, we know that G protein receptors can change cell behavior, right? So essentially that pathway, the thing that it's going to do is it's going to open ion channels. And opening ion channels is eventually going to start an action potential. So if we start that action potential, it moves along the axon of this neuron back into the olfactory bulb, and now we're actually moving towards the CNS, towards the brain. Do you have a question? Yeah. That's okay. Cat people. Oh, actually, no, but I do know what you're talking about. So, sorry for those of you listening on the podcast thing. No, it's okay. So she was asking about like the sort of specialized thing that cats can do that kind of addict us to cats, and that's actually a kind of I think it's a protozoan. So it's a species of sort of single-celled organism that they emit mostly through their urine, and then we breathe in, and it gets into our body and makes us like cats more. It's kind of it's a little, I know it's a little bit ridiculous, but yeah, no, so that actually does it. Yeah. But that being said, there is a lot of research on odorant like profiles in like people that we care about. We tend to gravitate towards people that have particular odorant profiles, and that's one of the things that they do is they essentially use a mass spec, which is a chemistry sort of machine that can analyze molecules in the air and say, okay, these people consistently have these smells, and these people are always attracted to those smells. So there has been some analysis of that in terms of relationships and family and how we relate to other people, because we like things that are similar. Generally speaking, but it's a little different. Not so much the cat. My cat has totally addicted me, so I get you. I get that. I didn't even like cats before Ray. So back to the basics here. So we're still on this first neuron. Our neuron has sent an action potential into the olfactory bulb. The olfactory bulb will So let's say the action potential moves into olfactory bulb, and at that point there is a synapse. This second neuron moves, oops, that's messy, hold on, let me go back. That part's convenient. Look at that, I can just erase things. Okay, technology. Okay, so the second neuron moves along that olfactory bulb, but the olfactory bulb is directly, and remember we saw this in our lab, the olfactory bulb is directly attached to the olfactory tract. So moves along the olfactory tract, into the brain, because remember a tract is in the CNS. Oh, yeah. Thank you. And then we're going to move to the olfactory cortex. And the limbic system. The olfactory cortex does what all cortexes do. It sort of processes that information. The limbic system is dealing with emotion and memory. Essentially the part of like smelling chocolate chip cookies that makes you think of being a kid. Smells are very closely associated with memory. For this particular pathway, there's no crossing over, which makes sense. I mean, the olfactory bulbs are really in the center of the brain, and it doesn't matter which direction that came from. It's not like with vision where you might want to know whether it's left or right. The smell's the smell, right? We're not following our smells left or right. Most of us aren't, at least, unless you're much better at smelling than I am. This is also one of the few senses that doesn't use the thalamus. So almost every other sense, including just touch, goes through the thalamus for processing, but this one does not. Any questions about those? Pretty straightforward, right? Okay. Let's see. And taste is just a little bit more complex, but not by much. We can pretty much say the same things are happening. I'm going to skip a tiny bit here talking about taste buds. That's what's mostly going on at this top part. So I'm not going to have you, I'm going to explain this, but I don't need you to know it for the test. I'm just going to kind of walk you through it. Essentially, you have your tongue. It's covered in all these little papillae. Those are all those little bumps that kind of get infected every once in a while. They hurt a little bit, right? So we have these different shaped papillae, and on the sides of those are where we have our taste buds. So the taste buds kind of look like this, and those taste buds have taste hairs on them, and the taste hairs have receptors. Okay? So that's the basic thing that's going on here. There's a little more detail. If you want to read about it, you can, if you're kind of curious about how those little cold sores happen, but that's all in your book. I'm not going to test you on it. Instead, what I want you to know is this part of things. So we have receptors on a taste hair. Okay. And there are two different types of receptors that we could have. They could either be receptors that open an ion channel, And those receptors are the ones that are over here. They'll either open a sodium or a H+ channel. So if they're opening a sodium channel, then that's going to give us the taste of salts. And if they're opening an H+ channel, that's going to give us, it's an acid, right? H+ is an acid. That's going to give us the taste of sour. Either way, those are positive ions that come into the cell, so they're going to depolarize the cell. Okay. So that's one type of receptor, ion channel receptors. The other ones are all G protein receptors. So they're going to start a signal transduction pathway. So these receptors on the taste hair start to send a signal. It could either be salts or sour, and then there are these other types of flavors. Okay, so I'm going to go through those 3 different types of G protein receptors. The first one is sweet. So that would be a G protein receptor that attaches to something like glucose or fructose, or even things like Splenda, sucralose. Fake sugars attached to those as well. You could also have alkaloids attach, and alkaloids are bitter. The most common places that we see them in our foods are in things like coffee or hops or tannins. So all of those vices that we're not supposed to drink, those are the things we're looking for there. Let me get this a little closer in here. Sorry, it says tannins if you can't read it. So coffee, hops, tannins. So essentially any of these alkaloids that we're drinking, almost all of them are associated with a bitter receptor, but when we drink them, we almost always associate it with, have it in the same thing as something sweet. It's just sort of how humans like it. There's the hypothesis about that is that alkaloids as a chemical are something we're technically not supposed to ever have because they are often found in poisons. And so most rats that are really sensitive to poisons won't eat alkaloids at all. But when we mix them with something sweet, then we like them a lot because it sort of masks that brain reaction that's like, it's also probably the reason you have to work your way into having coffee or really hoppy beers. You don't just like them from the second you drink them. You kind of have to work on it. And over time, you get more and more okay with it. But that's sort of a side note. You don't need to know that, but I think it's interesting. So alkaloids, we might think they're poison, but we like them a lot. All right, and then the last one is this sort of strange flavor that scientists didn't actually know existed for a long time. It's called umami flavor. Umami flavor in Japanese means savory proteins or something like that. It's known as the good flavor. So essentially meats have a particular type of protein that can attach to these umami receptors that give that sort of fatty sort of mouthfeel to the food that is a separate receptor that indicates umami flavor. It's kind of hard to describe, but if you're a meat eater, you know the flavor that I'm talking about, right? So it's just sort of a rich flavor that combines with other things. All of these also, as a sort of side note, when we're eating food, we also get touch receptors activated as well, and that makes a difference as to how we process food. This is just purely for the taste part of it, right? You know, when you're eating pudding, it feels different in your mouth than if you're eating bread or meat or whatever, and that sort of combines in our brain to process that information, both in our limbic system and then also in just our overall brain structure, the somatosensory cortex. Okay, so we activate these receptors. Either way, the receptors send an action potential. So I'm just going to say receptors activate and send action potential, right? So they have different mechanisms for doing so, but generally speaking, they're going to let some sort of ions, some sort of ions into that cell. It's going to depolarize the membrane. I'm just going to put depolarize over here so we remember. So they're depolarizing the membrane. You can see a picture of this over here. So you've got these, this is a hair cell. There's some type of receptor that's opening in here and depolarizes the hair cell. And then down at the bottom of the hair cell, we have a synapse, right? So we're going to essentially depolarize the cell, which is going to depolarize the membrane. Technically, that's considered an action potential in a hair cell, and we're going to have a synapse. And we'll say it's from the hair cell to the nerve. These are our cranial nerves. And we'll talk about which ones later. From there, we go to the medulla, and then cross over in the thalamus. So we go from the medulla, cross over in the thalamus, and then we go to what's called the gustatory cortex. Gustatory just means to taste. So medulla, thalamus, gustatory cortex. Not necessarily. That probably has more to do with the limbic system as well as memory of taste, and somewhat to the gustatory cortex, because you can train yourself to like something more by being more associated with it. There are some sort of, I don't know a lot of the details about how it works essentially, but there are a lot of sort of practical reasons why we might not like foods that we're not familiar with. As mammals, we have to be careful about what we eat. Our evolutionary history is not the same as our current history, where we can go to the grocery store and eat lots of things that we know are safe for us. Instead, there were berries that maybe we could try, but they might also kill us, right? And so a lot of those things are associations with like what we have in the past been successful at eating, and any time that we have a bad reaction to food, that can lead to a continuing bad reaction to that food, because we have a memory of it making us feel sick before, even if it wasn't the cause of the illness. So the liking or disliking thing is sort of a complicated thing when it comes to how our psychology of liking things doesn't work, or does or doesn't work. I've read in the past that there, for some children, when they're trying new things, they have to taste something new up to 40 times before they're really familiar with it, which makes sense, The first time I ate sushi, I wasn't sure about it, and then the second time I was like, well, maybe it's okay, and I had rice before, I'd even had seaweed before, but it was just a new combination of flavors. But by the same margin, like before I had my appendix out, I ate lettuce, and I literally couldn't eat lettuce for 2 years. Like, it just made me nauseous every time. I had to work into getting to the point where I could eat lettuce again, because at first I just thought I was sick from a salad that I ate, and then it got worse and worse and worse, and then it just got stuck. It was a terrible time. terrible time. I love phthalates, so you had to really work at that one. But, you just develop these memories of things because it's adaptive. It's good for you to remember when something made you sick, if, even if it's years in the past or years in the future that you might come across it again. All right, so basic pathway there. We're going to, are we going to do vision? Yeah, let's do vision. We can blast through this. We can do it. Okay, so vision, we're just going to talk about the basics of some of this anatomy of the eye that we didn't really talk about in lab, just because they're important. So we just need brief functions of these structures. Eyebrows and eyelashes are functioning mostly to prevent particles, liquid, and other substances from entering the eye. So we just want to prevent, any type of debris from getting into the eye if possible. Your eyelid also functions in that way. It can open and close. It can stop things from coming through. The eyebrows really act as a filter for particulate matter. Then if we happen to get any of those particles into the eye, then that's where the lacrimal apparatus works, and this has the function of creating tears. And those tears are there to flush the eye. So essentially over here we have the lacrimal gland, and this is where tears are coming from. Right? It's an exocrine gland. It's producing the liquid that comes across. And then as those teardrops are being formed, they're being pushed to the opposite side just because of diffusion. Right? So they're made over here on the lateral side of the eye, and then they're moving towards the medial side of the eye. And then on that medial side right by the nose, you have the nasolacrimal duct, and this is your system of drainage. So a lot of those tears are flushing through the duct, including with all that particulate matter, and they're moving into your nasal cavity where they can just be ejected either through blowing your nose or swallowing some of that mucus. So that helps us to get any of those substances out. It's why your eyes water when you're being subjected to onions, and it's just trying to get those chemicals off the surface of your eye because they're a little bit caustic. They don't feel very good. Exactly. When your eyes are watering, your nose almost always starts to run, and that's because you're filtering out whatever's in your eyes, and they start to produce more tears because of it. So, yep. We have a couple of visual reflexes. We're going to do those in lab tomorrow, so we're just going to worry about that. The snow is getting out of control. So I'm just going to write in lab. Okay. And we have the eye anatomy that we've already talked about, so I'm also going to skip that just for the time thing. So I won't ask you about any of the anatomy on this lecture exam because we've already been tested on it and you guys did really well. But we are going to talk a little bit about accommodation, which is how we see things through the eye. So accommodation, this is this top section up here. The basics are we know that our lens changes shape to focus. So this changes what's called our visual acuity. It's like how well we're able to focus, and we're going to test that a little bit tomorrow. So visual acuity is like how well we can focus our lens onto things. And as our lens if our lens has changed shape, then that can cause us to have reduced vision, or if we have weakening of the muscles of the eye, that can cause us to have a change in vision. So you have your lens right here. I'm not going to draw this on here, but I'm going to try and get it really big. So right in here is where that ciliary body is with the suspensory ligament, right? That suspensory ligament attaches the ciliary body to the lens. The ciliary body will then pull on the lens. It changes its shape, and that's how we focus. Okay, so that's the basics of what's happening here. When we have an object that's really far away, so this is us focusing on a house, then our pupil is going to be quite large because we're trying to look at something far away. The ligaments, our suspensory ligaments, are tight, and that makes the lens very long and thin. Okay, so that gives us distant vision. When we want to focus on something that's much closer, then our ligaments go very slack, they're loose. Our lens becomes short and fat, and that gives us near vision. When we saw our lenses, they probably were shaped mostly like that near vision, right? Because they weren't attached anymore to the ligaments, so they're very slack. Right? So that's the basic of what we're doing. We're pulling on the ciliary body, or we're loosening the ciliary body. If we're contracting it, then we have distant vision. If we're non-contracting, then we have vision up close. Okay. Any questions about that basic focusing? Okay. So what we are trying to focus is the light, and the light needs to focus on the retina. Okay, so the retina, the function of the retina, you guys keep looking out the window, and I'm like, don't look. Pretend it's not snowing that hard. It's fine. It's fine. So it's going to be okay. So the retina is that sort of loose, goopy layer that we saw the other day when we were dissecting our eyes. Full of nervous tissue, that's all it is. And the retina, in its entire function, is to convert our light into a piece. So we're just converting our light into an action potential, something that the brain can understand. Now, the retina coats the entire back of the eye. It's full of the vitreous humor. So essentially what's going to happen, and what we're seeing in this picture, is that up here on this top part, this would be the choroid. So this would be kind of the back of the eye. Whereas up here, this would be where the lens is, like way towards the front. So light would be coming by this direction. So it's coming from the lens up past the choroid, or up to the choroid. As that light is coming through the lens and up here, then these receptors, which are photoreceptors, we have one of them over here on the right, then these receptors are sensitive to that light. Right? So essentially this whole entire thing is the layers of the retina. These top parts are the photoreceptors that make up or are on the rods and cones of the retina. So the cells are called rods and cones. So that's really this top part. On mine it's purple. On yours it's kind of just gray. This next part right here, the middle part Mine is red, so it goes from up here all the way down here. Those are bipolar neurons. Oops, sorry. Bipolar neurons. And these down here are the neurons of the optic nerve. Okay, so we're just going to move over to this one detailed image. This is one of our rods. We really just talk about black versus white vision, so light and dark, and that's it. We don't really address color. Cones are really dealing more with color. So we're just going to focus on the rods. Rods contain a molecule called rhodopsin. Okay, so rods, let's make this work, contain rhodopsin. And rhodopsin is a really important molecule because when light hits rhodopsin, it falls apart. What it falls apart into are two different molecules. One of them is called retinol, and the other is opsin. Opsin is a signal molecule. Okay, so we have retinol, we have opsin. They're essentially making up this photoreceptor part of the eye. Falls apart when light hits it, so it's together in the dark. And this next page is showing you two different options where we either have light or we don't have light. So on this side, we'll say this is in the dark. And then these arrows are in the light. Okay, so on the right, we have in the light. On the left, we have in the dark. Now, the tricky part about vision, the thing that we have to remember about vision, is that it's kind of the opposite of most other things. When we have odorant molecules, then we start a signal because something attached, right? When you have touch, you start a signal because something touched you. In this case, in the dark, we have constant action potentials. In the light, the APs are stopped. But these are all individual receptors, right? So when I'm looking at things that are light or dark, like in this room, there are some parts that are darker, there are black areas, there are white areas. Then we have different receptors that are getting different amount of light. They're not necessarily all going to be light or all going to be dark, unless you walk into a bright space or a dark space. Okay. So essentially, in the dark, when our eyes are closed or when we've just started hitting black absorption of light, when I'm looking at dark things, there's constant action potentials. The reason there's constant action potentials is that we have rhodopsin together. And that means that we have sodium channels open. which means that this cell is depolarized. When the cell is depolarized, we can release neurotransmitters. And so then essentially we have an action potential in those bipolar neurons, and we have action potentials down here, heading towards the brain on the optic nerves. So right here, this is a synapse. Right here, there's a synapse. So we're essentially doing just full signals through 3 different neurons. Well, through one rod, then 2 neurons. Okay, so this is all happening in the dark. Constant action potentials. Lots of neurotransmitters released, moving along that optic nerve, getting constant signals. But then when light hits, this retinol plus opsin reaction happens, and the opsin, as a signal molecule, attaches to a G protein receptor. So if this was a receptor, then right there on whatever's left of that retinol molecule, the opsin attaches, G protein receptor, we have signal transduction. So we go through that whole adenylate, cyclase, protein kinase, etc. Signal transduction, and our signal transduction will close the sodium channels. This causes the cell to repolarize. Repolarizes, and we have less APs on the optic nerve. So we've just reduced the action potentials on that nerve. Does that make sense? Okay, good. So then we're just going to do this pathway right here. Okay. So the basic pathway we're looking at is starting up here. All right, this is the light, or the direction the light would be coming. So in the dark, we'd be sending action potentials through that retina, through the optic disc, along the optic nerve. Right? From the optic nerve, we'd be following along, and at this point, there are 2 options. We're either coming into the optic chiasm and crossing over, and I'll write this out as a pathway in just a sec. So we're coming into the optic chiasm and crossing over, or going straight back into the thalamus. Okay? So some of the signals will cross over, some will not. If we're going to the thalamus on either side, depending on whether it's contralateral or ipsilateral, and then from there, we go back to that occipital lobe to the visual cortex. Okay? where the signal's processed. The reason that we have some signals crossing over and some not is that gives us some depth perception, right? So we can get some idea of the size and how far away objects are based on which neurons are firing, whether they're crossing over or whether they're not. It helps us to put all that information together to get a more three-dimensional image. Okay. So we already saw the optic chiasm. We saw what that looks like. You know that pretty much after this point, we become nerves because we're moving back into the brain, right? Okay, so I'm just going to write this out, and we're actually going to skip this bottom section here. So I'm just going to write that like a pathway so that we have it in writing as well, just so we, you have something to study with. So actions, potentials, we'll just start from there. APs start on the optic nerve, so that's after all of that rhodopsin stuff. Okay. The APs will either cross over in the optic chiasm or go straight to the thalamus, where they'll synapse. And then from there, they'll head to, so that second neuron moves AP to primary visual cortex on the occipital lobe. And then just to make a note of this, this is because it allows depth perception. That should be a C. So it's the reason my cat, who only has one eye, does not have any depth perception and sometimes runs into the wall. It's really funny, actually. He's very cute about it. I kind of want to bring my cat for you guys to meet because he's so great. Okay, good. So we're doing ears tomorrow. So we're going to skip this ear part because we're doing that tomorrow, but we are going to talk about balance, and then we'll be done. So you might get out a couple of minutes early because I know the snow is looking terrifying. So don't worry about pitch and loudness. We'll talk about that tomorrow. We're just going to do balance starting at 8. Hi. So these are two structures in the brain two structures in the ear that we'll talk about tomorrow, or that we'll see tomorrow on the models. These big sort of loops are called the semicircular canals. And then this whole area right here, sort of in the center, is called the vestibule. The vestibule. This sort of shell-like structure, that's the cochlea, that's for hearing, it's not for balance. These two are both balance structures. So essentially, you have two different parts of balance, or how we figure out where we're positioned in space. And those two parts are either coming from the vestibule or the semicircular canals, where the vestibule is giving us information about head position, and the semicircular canals, so let me do, just to make sure it's clear on your notes, so the vestibule, if I can spell vestibule, okay, the vestibule gives us information about head position, and then the semicircular canals are giving information about rotation and acceleration. Okay, so essentially what's happening in either of these situations is that we have these hair cells. Okay, so you can see the hair cells right here. You can also see hair cells up here. And when those hair cells bend, so hair cells bend, then mechanical receptors send a signal. They're actually bending, and then sodium comes into the cell. I'm just going to say sodium influx. Right, so they're going to end up depolarizing that cell because of the sodium influx. The hair cell depolarizes and then synapses to the vestibular nerve, which is sort of a joined nerve. It's one of our cranial nerves called vestibulocochlear nerve. So we'll see both of those nerves tomorrow. Okay. So let's just go back. So this is the basic. Hair cells bend. Mechanical receptors send a signal. Sodium influx. Hair depolarizes. We synapse to the vestibular nerve. That vestibular nerve then goes back to the brain. Okay? We send balance signals to a bunch of different places. The cerebellum is one of those places. The primary motor cortex is one of those places. We need information about balance in a couple different ways. So we're not going to worry about the rest of the pathway. We are going to just mention a little bit more about how those hair cells bend, because it's a little different in each of those two structures. Okay? In this structure, you have this entire sort of flame-shaped covering over the hair cells. It's called an ampulla. And the whole ampulla can shift as we move. So as we shake our head, or as you get into a car and you start to accelerate, those entire ampulla move, the hair cells bend. This is one of the reasons that people can get car sick, because their acceleration isn't matching what they expect to be happening. You're not actually moving, but your car is telling you you're moving because of the acceleration, right? So your vision doesn't match with your semicircular canals, and that's complicated, right? Your body starts feeling a little off. In the vestibule, we have these other structures. They're called maculae. Okay. The maculae are in these little guys called the saccule and the utricle, okay, where the maculae are this sort of big structure here. There are two C's there. You just can't see them. Okay. So the maculae are made up, down here you've got these hair cells that have these receptors coming up, all right, the little hairs. There is a gel that's right here, and then on top of this gel are rocks. They literally are a type of rock. They've got calcium in them. And the rocks, when we shift, our rocks move. Okay. The rocks do have a name. They're called otoliths. Essentially, they roll from one side to the other. They should be kind of embedded within that gel. So as they shift, the gel shifts, and then the hairs bend. In some older people and in just some genetic cases, those otoliths can get loose, like the gel doesn't hold them down and they sort of move around even when someone's staying still. And that can cause what's called vertigo, so someone that feels like they're moving when they're not actually moving. And there is a kind of interesting maneuver where you can shift those rocks into a position so they can't roll around anymore. It's like a series of, I think, 16 different head positions that you have to go in a really specific order, but it can relieve vertigo for a pretty long period of time, so it can actually be really helpful. Any questions about that? Okay, that's all I've got for balance. So I'm going to let you go off into the world. Tomorrow we'll talk about ears. And I hope you have a safe drive home. Go really slow, okay? Be careful.